is Lloyd, and I'm the research ecologist at the Southeastern Center for Conservation here at the Atlanta Botanical Garden. And I'll be your host this afternoon. Before we begin, I'd like to remind you all to make sure your phones are switched to the silent mode. Today, we are honored to have with us Dr. Donna McDermott with her talk titled Peer Pressure and Pesticides, How Bees Choose Flowers in Complicated Ecosystems. First, let me tell you a little bit about Donna. Donna studies the behavioral ecology of foraging bumblebees. She recently completed her PhD in population biology, ecology, and evolution at Emory University. Donna and I actually used to work together in the same lab. We had the same PhD advisor. This fall, Donna will be beginning a faculty position in Emory's English department as an assistant teaching professor, teaching science writing. She has previously taught courses in animal behavior, science communication, interdisciplinary writing, and pedagogy. In addition to her work as an educator, Donna has also worked as a journalist through the AAAS Mass Media Fellowship, a wildlife biologist, a program coordinator for the Science Atlantis Community Science Outreach, and also a ghost tour guide. So without further ado, I'll give the floor over to Donna. Please give me applause for Donna.
Hi everyone. Hi everyone, and thank you, Loy, for that introduction. Can you hear me? Yes. Great. So I put the word flowers in my talk, but that is really just a way to sneak into this garden. Most of this talk is going to be about bees, which I'm excited to talk about with you today. I have asked that everyone please wear a mask throughout the talk. I can't enforce that in any way, but I do have some underlying health conditions, and I've discovered during this season of our world that I really don't want to die. So please help me out with that. So Loy mentioned that I am now teaching science writing, which is exciting for me. It's my dream job. But I realized as I was putting together this talk that I can't phone this stuff in anymore. If you are a scientist and you have a spelling or a grammar mistake or something like that, people can sort of say like, well, she's probably good at math. But I've lost that barrier of protection. So luckily, I've had a good number of conversations about my research and have had some practice in getting it right, because those conversations can easily go wrong. For example, if I'm in an Uber getting my hair cut or something, some new acquaintance will ask me what I do for work, and I'll say, oh, well, I'm a biologist. And they'll say, oh, wow, are you doing something like finding a cure for a disease? And I'll say, no, I study bees. And generally, they will tell me that, oh, I've heard that the bees are dying. Is that true? And then I say yes, and then we stare at each other for a long time. Or with that conversation can be slightly more productive, right? If someone's asking me about the bees and how they're doing, I can say, like, well, yes, we have some general information that bees' populations are declining. We know that that's happening because of a range of different factors, both environmental and human influenced. Most of our information is about honeybees, which are actually more of like a managed livestock species. In reality, there are thousands of species of bees all over the world. Most of those wild species are great pollinators, and we have less information actually about how they are doing, though we have reason to suspect that their populations are also declining, which in terms of human well-being and our ability to access fresh fruits and vegetables is pretty catastrophic. So then we enter the silence where we stare at each other. But in this scenario, our silent despair is more well-informed. And much as that kind of doom and gloom is really sucks you in, it's really not accurate. There is not a death sentence hanging over bees, at least not a mandatory one. We know plenty of strategies for preserving their habitats and the flowers that they need to survive. We know plenty of strategies for ameliorating climate change and for conserving ecosystems that we rely on for our own lives. We know that these strategies involve preserving wild spaces, creating structural supports for people in our society, and also creating economic systems that don't rely on constant growth and expansion. And scientific research, like what I'll be talking about today, certainly plays a role in working towards a sustainable future for bees and other aspects of our environment. But really, the more important work for saving the bees is being done by environmental justice activists, like, for example, in our own city, the Defend the Atlanta Forest Initiative. These folks are literally working day or night to conserve natural forested public land as a community resource. Work like this that addresses immediate environmental concerns and racial justice are essential for being able to do things like scientific research, which also works towards environmental solutions, but is slow and is looking for those more long-term strategies. So if you do support science, I ask that you check out this initiative, consider contributing perhaps time or money, because it really is essential for scientific work. Because of the activism like this, I can give this talk today, in which I'll be focusing on some of those bigger, deeper, long-term questions that we have the privilege of exploring here. First and foremost, what is actually happening with all of those bees? So as some background, bee foraging is a fairly involved task. Bees are visiting flowers over and over again throughout the day, possibly for most days of their lives. They're collecting nectar and pollen, not only for themselves, but for possibly hundreds of their sisters. They need to remember which flowers offer the best pollen and nectar rewards in an environment where the flowers that are blooming are constantly changing, so they need to update that information throughout the growing season. They're figuring out how to evade competitors and predators and parasites, all while managing all of the, and understanding how to manipulate the flowers in order to find the rewards within. It's a lot of information, and it is essential to us, since inadvertently, as they are foraging, they are also pollinating our crops and the natural flowers that, the wild growing flowers that are the basis of the ecosystems we rely on. And as I mentioned, bee populations are declining in number. So there are a large group of scientists racing to learn more about bees so that we can better understand how our natural environments are changing. So what I'm going to be telling you about today is about how my research explored how bees make choices about which flowers to visit, and how those flower choices are complicated, both by the presence of other bees, here described as peers, and by the environmental impact of pesticide exposure. And I'm actually going to start this by telling you our big results, which is a bit of a spoiling of the ending. But I want to be able to go back and explain how we got to those results with more of a focus on experimental design. So in particular, I found that when bees are influenced by their peers, they learn best when they get incorrect information about the flowers around them. And in terms of pesticides, I found that bees ignore their peers after they are exposed to pesticides. So how did we get there? To give you more of a setting, all of my bee research was done in the lab. Loy mentioned that he and I worked in the same research group, which is fun. This is one of his pictures from his research days. He worked in the Rocky Mountains in these beautiful wildflower meadows. I did a lot of my work in this closet. We hung red lights so that the bees wouldn't be disturbed when we went in there, and we kept the bees in these series of boxes. So we were studying bumblebees, and the colonies that they make look more like this. Each of these sort of pots is about the size of a bumblebee. It's not the structure you're used to seeing with honeybees. The stuff you're seeing in the pots is technically honey, but you can't extract it the way you can honeybee honey, so I wouldn't go ahead and try to eat that. And instead of studying them in a meadow, we had an artificial foraging arena, which is the technical term for what really was a robot meadow. These flowers were 3D printed, and we dispensed artificial sugar water through, it was regular sugar water, artificial nectar through those fake flowers. That nectar came through a series of tubes that ran underneath our fake meadow and up through the flowers, after being supplied by an elaborate group of reservoirs where either sugar water or water came down through. The bees themselves triggered the release of sugar water after we tagged them. So this is a device that I use to glue a tag to a bee. This is an RFID tag, that is radio frequency identification. It's basically a little radio antennae that we glued onto their backs. So when they visited a flower, it triggered the flower to release a droplet of fake nectar that the bee could then go and drink. So we had automatic information about which bees were visiting which flowers, and when and how often. And we could even trigger the flowers to dispense nectar droplets in certain ways, like at certain probabilities or at certain timings, so the bees couldn't just sit there and continuously drink. We also, to understand the influence of a bee's peers, we used a model bee that we pinned onto the surface of a flower. If you're really looking, what we are calling a model bee is actually a dead bee on a pin. There's a considerable amount of previous research that uses these dead bees on pins as experimental techniques. It seems that the bees don't notice that they are following like a dead peer. It's a little morbid, but they don't seem to care. And we were interested in what they would do. If you're just walking in, I have asked that everyone could please wear a mask while they are attending the talk. There's some on that table over there. So, the technical term for this influence of peers is called a social cue. We already know, this information that a bee is getting from another animal is, in this case, the sight of another bee on a flower. That is what a social cue is in this context. And that tells the bee that the flower is likely to be rewarding, so that they go and visit it. Bees make decisions about which flowers to visit using both these social cues and their own experience of trial and error. We already know a little bit about social cues and learning in animals. For example, this study, researchers looked at how pigeons who follow other pigeons to find food. They found that the pigeons who follow others, who follow those social cues, are not able to find that food on subsequent trips without social cues. So they can follow someone, but once they've got to do it on their own, they're all confused again, until they go and figure it out by their own trial and error. However, in this study, following social cues was reliable and an easy way to get information about where to find food. So why would the pigeon be doing all that work to figure it out themselves when they could just follow someone else? In my study, I was interested in the idea of misleading social information, or social cues. So if these bees, like these pigeons, don't learn from following social cues, perhaps they would be more influenced by misleading social cues, wrong information. So how would I study this? Well, the first step is to design an experiment that tests learning. And that's harder than it sounds. In these sorts of animal learning studies, you need to anticipate for the fact that once you test an animal, they are still learning from the test. So, to give you a more concrete example, in my study on one day, I wanted to teach bees that blue was good and green was bad, as in a blue flower gave them a sugar water reward, and a green flower only offered water, which is not particularly a reward. And I wanted to involve social cues in that as a way to lead them to that good blue flower. So if bees correctly learned that blue is rewarding on day 1, then what would we expect to see on day 2? If we put the bees back into the foraging chamber again, our robot meadow, and we gave them a similar scenario to forage in, how would we know that they know blue? Depends on how we set that up. So if we gave them the same scenario, that wouldn't work. And I'm going to explain why, and then go through a couple of other things that don't work in order to justify what we did do. So if we gave bees the same scenario on day 1 and day 2, and we found that bees made the majority of their visits to the blue flower, that could mean that they learned that blue was good on day 1. Or it could mean that they learned that green is bad on day 1, and nothing about blue. Or it could mean that they didn't learn anything on day 1, and they're just now learning that blue is better, so they're preferring to visit it. Alternatively, if bees visited the green flower more often, that wouldn't mean anything to us at all, so we wouldn't have anything to do with that data. And as an experiment, this doesn't work, because there are multiple explanations for the same behavior. We wouldn't be able to gather any new insight into what bees are doing. And it's not really much better if we make small changes. Like, we could make that green flower also now rewarding, and that would change our options a little bit. Now, if bees are visiting the green flower, that might be telling us that they didn't learn that blue is better on day 1. Or it could mean that they did learn that blue is better on day 1, but it's no longer like better, so they don't care anymore and they're just visiting whatever they see. Again, multiple explanations, same behavior, it doesn't work. If we make that blue flower not rewarding, we still have multiple explanations. If we make the green flower not rewarding, we still have multiple explanations in this particular setup. And also, we enter the possibility that the bees will just stop foraging because there's nothing in it for them. These bees are obnoxiously clever for the purpose of experiments. They keep learning stuff about the flowers and how they need to orient them. Some bees will figure out how to jam themselves in a flower so that it just floods out nectar. We have to figure out how to avoid that for them. It's endearing, but very annoying in the moment. So we had to get rid of that green flower, and we replaced it with a totally new flower, a purple one, that they wouldn't have any association with. And here, they are both rewarding, so that our best explanation for each of these behaviors would be that if bees predominantly visited blue, that would mean that they learned that blue is better on day 1. And if bees did not predominantly visit blue, that would indicate that they didn't learn that blue is better on day 1. All right, so that's settled. But we're not done, because we need to be able to distinguish between whether bees learned something about the color of the flower or the fact that the social cue was there, which is a major question in this research. So we need to change up what we're doing on day 1. Not only do we want to have these honest cues, we also want to have misleading cues, which is a social cue leading the bee to a bad flower, and no cue at all as a control. We need to be able to know, we need to be able to compare misleading cue bees to honest cue bees to the no cue bees. So that way we can control for things like inherent preferences for different colors, or whether bees just like novelty, or whether there was a weird energy in the lab that day and they all decided to not forage, or something like that. All right. Sorry. Got ahead of myself. So that is our experimental design. Bees on day 1 were presented with one set of flowers. Day 2 presented with a different set of flowers. They went in and foraged by themselves on each day for 30 minutes in each of these setups. And here's what we found. In this graph on the long x-axis, you're looking at the 2 different days of foraging, day 1 and day 2. And on that tall y-axis, you're looking at the proportion of visits that bees made to blue flowers. So in all of their visits, how often were they going to blue? Again, on day 1, they had the option between a blue and a green flower, and on day 2, a blue and a purple flower. We compared the behavior of bees that were trained with no social cue, an honest cue, and a misleading cue. And this is not data. This dotted line is right at 0.5 to show us what it would look like if bees had no preference for either flower. And here's what we found for bees that trained with no social cues. They showed a small preference for the blue flower on day 1, which makes sense, it was better. and no preference for either flower on day 2, which again makes sense. They were equally as good as one another. But this is consistent with what we would expect if bees did not learn that blue is better on day 1. Similarly, for bees trained with honest social cues, they showed a strong preference for that blue flower on day 1, and then no preference for either flower on day 2, again consistent with not remembering the blue flower from day 1. This blue point on day 2, it's not quite on the line, but it's not significantly far away from it. And finally, for bees trained with misleading social cues, they did not show a preference for blue flowers on day 1. They visited that green flower, they followed their social cue to it over and over again, even though it didn't offer them anything. But eventually, they figured out what was going on, so that on day 2, they visited the blue flower more often. Again, consistent with what we would expect if they remembered it, because they learned it in the first place, that blue is what's good. These faint dots are the average for each bee in each group. So you can think of the little dots as glomming together into one of those big dots. If you're into this kind of thing, those lines are 95% confidence intervals. Okay. So in result, we found that bees followed social cues while they were present on day 1, but then once they were gone, bees only learned about flower color with misleading social cues. So I would compare this to my own experience of navigating someplace with a GPS. I know that if the GPS is on and it's giving me information about where to go in the city, I will definitely follow those instructions, and they're acting like a social cue. They're telling me what information I need. But I found that I'm not very good at learning how to get somewhere if I've only ever gone there with a GPS. If I really want to know where something is, I have to turn off my GPS and kind of figure it out and go down the wrong road and take a turn and have to double back and figure out how these streets all connect to one another. Then I learn where some place is. Similarly, our bees don't learn that association between blue and reward unless they are training with misleading information that they have to overcome. And someone could take this research and keep going to explore which environments bees are more likely to learn in. Under what circumstances are bees doing learning? It's not a neutral act. For us, I mean, we do it all the time, but especially for such a small animal, learning takes energy, it takes time. But perhaps bees are more likely to learn in environments where they are foraging with a bunch of other species of bees. Those other species are more likely to give them incorrect social cues. So perhaps that's better for learning than an environment where they're foraging with just their own species. Or perhaps bees are doing more learning in an environment with diverse flowers so that they are learning different things about how to access the flowers and which ones are blooming, as opposed to, say, a crop field with like 1 million of the same plant. These kinds of real-world circumstances were interesting to us after developing this first experiment. So we decided to look at just one level of real-world reality with exposure to neonicotinoid pesticides. We wanted to know how that influenced bees. So in our question about how pesticides influence bees' use of social cues, their memory of flower color, we were studying in neonicotinoids. Neonicotinoids, or neonics, are the most commonly used class of insecticide used in the United States, and they're all over the place. They are used for crops or sometimes like people's front lawns, but they persist in soil and they travel through water, so they spread everywhere. And a wide body of research has shown that they have detrimental effects on mammals, on birds and amphibians, and on non-target insects like our bees. The companies that create these pesticides have to evaluate how much of the pesticide can be used before it straight up kills honeybees, and then recommend that that amount is used. But again, broad body of research showing that even at the appropriate amount, these pesticides still have effects on bees' behavior, reproduction, and other facets of how they live their lives. In particular, the facet that I'm most interested in is the effects on how bees learn. We know that neonics do affect how bees learn. For example, in this study, researchers evaluated bees' ability to navigate this brown thing is like a maze, where on each of those little blue platforms, there's a sugar water reward. So a bee that is unexposed to pesticides can go from the middle to each of those blue platforms to get a little bit of sugar water. But a bee that has been exposed, they aren't as good at remembering which platforms they've already been to, so they just revisit the same ones. It's not efficient, and they stop getting the reward after a little while. And that's not too surprising that they have these effects on working memory. Other studies have evaluated how neonics affect the developing brain of a bee and found that they inhibit growth in the part of the brain that does multisensory processing, so like getting different information through different senses and putting it into one coherent message. And that does affect their ability to learn later on in life. So we have this broad body of research telling us that neonics are bad for how bees learn, but I was interested in how bees might compensate for that lack of ability to learn. Like maybe, getting ahead of myself. So for example, we know that bees are more likely to use social cues in challenging tasks. Well, that sucks. It's not this. That's fine. Thank you for waving a hand. I would have been doing that for a while. Anybody seen any good bees lately?
Yeah, so we've got this. Okay.
I do have one question. I think it just.
Do you have a question?
When your talk discussed what kind of mosquito control is feasible without harming pollinators?
So for the recording, the question was, will my talk discuss what kinds of mosquito control are effective without harming pollinators? The answer is no. Sorry. Oh, yes, sorry.
Can you address color perception in bees so you know that blue, green, and purple are defined by bee ring?
Yeah, fun question. So the question was, can I address color perception in bees so we know that blue, green, and purple are like perceptible to them? The answer is yes. So other researchers have looked at the color space, is what they call it, of what bees can see. And it is shifted towards that blue and purple range. So you may have heard that bees can see ultraviolet, which is part of the light spectrum that we can't see. They see it as an additional color. If you haven't looked this up before, I would really recommend like UV flowers. Google that when you get home. Lots of flowers have like patterns. Oh, thank God, it's up again. Leading the bee into the part of the flower that has the nectar. So yes, they can see blue, green, and purple. They can't see like red so much. All right, so I was talking about what we already know about social cues and learning. Okay, and we know a little bit, oh, right, okay. So in this study, researchers looked at bees' use of social cues in an environment where they were distinguishing between flowers that were either easy to distinguish between or hard to distinguish between. And they marked their flowers not by color, but by a little sign with a number of lines on it. So it was easy for bees to distinguish between flowers like these, which were marked by either 5 lines or 2 lines, but much harder for bees to distinguish between, say, 4 lines and 5 lines. And when they were in that harder to distinguish scenario, bees were more likely to rely on those social cues, follow them more often. So, perhaps if, as I was saying before, neonics have this detrimental effect on bees' ability to learn and remember, maybe when bees are exposed to neonics, they end up relying on social cues more in foraging. Perhaps that's how they compensate. And Before I show you what we found, let me talk you through what our results would look like if they either supported or disproved my original hypothesis. So again, in this graph, on that long y-axis, like in the last graph, we're looking at the proportion of visits that bees made to blue flowers. But I've switched up the x-axis, so now it is the 3 different social cue treatments. Again, this 0.5 line is going to be showing us what no preference would look like. And I'll be using color to distinguish between bees that were not and were exposed to pesticides. This information is actually coming from our last study. We already know it, that while social cues are present, bees that are not exposed to pesticides show a small preference for blue flowers. Bees with honest social cues show a strong preference for blue flowers because the social cue is leading them there. And bees with misleading social cues follow that cue to the green flower. If my hypothesis was correct, then we would expect this pattern to look more extreme for exposed bees. Not for these guys, because they didn't have a social cue. But here, honest cue bees should be following that cue to the blue flower even more. Misleading cue bees should be following that cue to the green flower even more. And if I'm wrong, then we wouldn't expect that more dramatic effect. We would expect all groups of bees that are exposed to pesticides to forage as if the social cues aren't there. All the same. And here's what we actually found. Again, here are the bees that are not exposed to pesticides, showing their slight preference, strong preference with the honest cue, no preference with the misleading cue. Here's the per bee data for all the bees in each group. And for bees that were exposed to pesticides, they showed no preference according to their social cue. Influenced by their social cue. They all preferred those blue flowers a little bit, and the same amount as one another, indicating that after we exposed bees to pesticides, they stopped following social cues. They ignored their peers and just visited the blue flower as if their peer wasn't there. And there is their per bee data. So my hypothesis was wrong. Bees that are exposed to neonicotinoid pesticides stop following social cues. It's a little bit like, I can never tell if this is like an appropriate analogy. It's a little bit like you've got a drunk person and you're trying to push them towards back into their bed, back into their house, but they keep wandering away somewhere else and it doesn't make sense to you, but they're no longer making decisions as you think they should. That's what our bees were doing. And this actually has strong potential implications for services, things like crop pollination. So to give that more context, this is someone else's study, it's not mine. But Dara Stanley and our colleagues studied bee pollination of apple flowers, and they found that when they followed just one bee foraging by itself, one bumblebee, those that were exposed to pesticides were just as good at pollinating apple flowers as their unexposed other bees. But when they compared groups that were all foraging together, either not pesticide exposed or pesticide exposed, they found that the unexposed groups were much better pollinators than the exposed groups. So this indicates that there's some sort of emergent benefit of foraging in a group for these bees, but that benefit is lost after they're exposed to pesticides. And hey, we found that social cues are lost after they're exposed to pesticides. So this is a bit speculative, but perhaps what bees are losing is the amount of attention they pay to one another. So they become worse at finding flowers or noticing which flowers have recently been emptied out, and they become worse pollinators. In fact, if a researcher wanted to, they could pick up where our study left off and examine how different groups of bees, pesticide exposed and not, communicate with one another using these social cues while they are out foraging on, say, a crop like apple pollinators. In particular, in this diagram, that's an outline for a study in which A researcher would train leaders and followers. A leader being a bee that has already been exposed to the environment, followers would be brand new. And seeing the results in pollination of groups that are either all exposed or have a pesticide-exposed leader or just a pesticide-exposed followers or whatnot. Yes. So as a final thing, I just wanted to say thank you to a few groups of people. The lab that I worked with, in particular, Loy, for inviting me here today. Some of the folks that worked on that robot meadow I talked about. I'm not an engineer, so the engineers who did, I'm very grateful for. And some of the many undergraduates who were really invaluable in doing all of this work. I've built in a lot of time for questions. So in returning to either this idea about how bees are influenced by their peers or how they're influenced by pesticide, what questions can I answer? Oh, thank you. Yes?
Is there also research in the agricultural field that correlates reduced productivity with pesticides?
So the question was, is there also research in the agricultural field that correlates reduced productivity with pesticides? I mean, that study that I showed you was taken from real apple orchards, which is the closest example I have off the top of my head. I can't give you a very well-informed answer right now, because I think the answer is yes, but I don't want to just say that because I just went on this whole pesticide spiel. I do know that when we get into actual agricultural fields, this kind of research gets a lot more complicated because there are generally lots of forms of pesticide, like an insecticide and a fungicide and an herbicide, all interacting together on bees, as well as other things like antibiotics. One of the researchers in our lab studied the effect of antibiotics on bee learning, which are also detrimental. So it's kind of a minefield out there for them. So being able to say like, oh, it's the insecticide is a little bit challenging. Yes?
Were the model bees used as just a visual cue, or did they have any pheromones or anything else for the other bees to be attracted to?
Great question. The question was, are the model bees used just as a visual cue, or do they have any pheromones or other types of cues that affect bee foraging? And the answer is that for our study, it was just visual. People have done work with those pheromone cues, but we actually specifically controlled the experiments to get rid of them because we weren't specifically looking for them and we didn't want to like stumble upon them by accident. So we wiped down all of our surfaces with alcohol to disrupt any potential scent cues left either by previous bees or by those model bees. I mentioned that those model bees are real bees, but they were dead and they have been frozen, which was another way that we prevented those pheromone cues or other sorts of scent cues.
Do the bumblebees dance like the honeybees to tell the other bees where flowers are?
The question was, do the bumblebees dance like the honeybees? And they don't. They actually don't really communicate with one another very much when they're in the hive about where to find flowers. And once they are out foraging, they're looking at one another. But I'm not aware of there being a preference for like looking at a nest mate versus looking at another bumblebee from a different colony or even a different species.
So social cue is just following another bumblebee?
Yeah, it's all visual. I mean, they do also follow pheromone cues and even like electrostatic cues on the flower. But that just wasn't what we were looking at in this study.
So would you say that, at least in this controlled experimental environment, that an individual pesticide-exposed bee, despite losing the ability to follow social cues, still made better decisions? It seemed like it kept visiting.
Yeah, that's an interesting question, right? It seems like bees should be punished for following these, for being exposed to pesticides in some way. And I was interested in looking at the just amount of sugar they were able to get while they were foraging to see if that differed after pesticide exposure. And in this setup, which again is very simple and straightforward, we didn't see that. The bees that were exposed to pesticides typically got just as much sugar out of the chamber as their unexposed match within each social cue group, except for these honest cue bees, right? Following an honest social cue was, marginally better, this is a trend, not a significant difference, than ignoring an honest social cue, which makes sense. In terms of the individual bees in that study with the apple flowers, it seemed like they were doing just as well also. But yeah, I can't speak more about that. Yeah.
So what are you going to do? What will you do with this knowledge now? How is that going to help?
What will I do with this knowledge now is the question. Oh man, I sort of have a cop-out because the answer is kind of nothing. I am no longer, in terms of research, I'm no longer doing bee research. I've transitioned over into studying education, which kind of is an answer to your question. That I realized I was more interested in learning about how people use information like this, how they pass it on to one another through teaching or through, say, journalism, rather than doing more of the field work myself.
Were you funded by anybody when you take this research and continue it?
I did this research with a graduate fellowship through the Department of Defense. So good question. Will other bee research be funded in that way? I have no idea. The lab that Loy and I worked in still studies bees. In particular, they study bees in nature and which flowers they visit and how that is affected by climate change. So certainly aspects of this are definitely going to be taken up. And these studies are going in for publication where people can adopt them into their next generation of studies.
So why is the DoD studying bees?
God, who knows? I wrote a fellowship application about food security and the future of our country, and that worked in 2015.
Okay. Thank you so much, Donna. Can everyone give a round of applause? So thank you everyone again for coming out tonight. Just a reminder that this talk is recorded. So those who have friends or family who are unable to be here today, they can watch this online. I'll show you the website in a second. This Science Cafe happens every third Thursday of every month, so join us again next month. If you enjoyed the talk today, next month we will have a speaker, Dr. Braddock, who will be talking about restoring the American chestnut to the southern Appalachian hardwood forests, which I think is very excited about that too. Just to know, just to know, it says here that the event is at Day Hall, but next month the event is actually going to be at Merchant Hall, which is across the way on the other side of this thing. So this is the email that you should be sending your questions or suggestions to our Improving Science Cafe. Send them to mglobal@atlantadg.org. This is Maria's email address. And if you want to know more about our department, check out our website at atlantadg.org/conservation-research. And you can follow us on Instagram